It's my pleasure to introduce Sean Lennox. He's a PhD student at Michigan State University's Aquatic Animal Health Lab and advised by Dr. Tom Locke. His research focuses on developing methods for preventing the vertical transmission of the causative agent of bacterial cold water disease, Flavobacterium psychrophilum, which causes substantial losses in trout and salmon facilities worldwide. So thanks for being back this summer, Sean, and presenting your work.
Of course, thank you. So before I get going, I want to first, of course, thank my co-authors for all of their contributions on this. So all of this work revolves around Flavobacterium psychrophilum, which is this yellow pigmented gram-negative bacterium shown in the image here. And like Nick just said, this pathogen causes some really substantial problems throughout hatchery and aquaculture facilities across the world. So today I'm really excited to share with you some of my work that's directly aimed at preventing a major transmission pathway of this pathogen, which is vertical transmission. So I know that I'm preaching to the choir with this, but I want to highlight the importance of aquaculture both globally and in the US before really getting into it. So aquaculture is the fastest growing animal agriculture industry globally. And less than a decade ago, we primarily obtained fish for human consumption through capture fisheries, which is shown here in the gray line. But since about 2015, we've actually been obtaining fish as an important human protein source, primarily through farming, which is shown in blue here. So it's just projected to keep on growing. And this industry is also is growing in the US, where we especially raise trout. So here we see that in 2019, we tried to raise 79.2 million trout, but we only ended up actually selling 47.2 million of these. And disease plays a really big role in this. So in 2019, we lost over a third of all trout produced through disease. And among the causes of these losses, pathogens within the family Flavobacteriaceae are a really major cause of this, and especially Flavobacterium psychrophilum. So like we already said, this bacterium is the causative agent of bacterial cold water disease, which in severe cases, it can cause tissue damage all the way down to the bone. And if it goes untreated, it can cause losses of greater than 70%. So there are a few different reasons why this is such a big problem. One is that it's able to be horizontally transmitted from infected fish to uninfected fish. And we know that dead fish especially can spread a lot of psychrophilum. And we're also seeing reduced susceptibility to the limited approved antibiotics that we have. And we don't have a commercially available and fully efficacious vaccine against the pathogen for use in the US. So to make matters even worse, on top of all of that, psychrophilum is one of the few pathogens we know of that's really efficiently able to be transmitted vertically, so from the parents to the offspring via infected reproductive fluids. So preventing this route of transmission, vertical transmission, is really the focus of all of my research. So some of you might remember, but last year I shared with you my work into developing a novel diagnostic assay to screen for psychrophilum under field conditions. And so that's this LAMP assay. So development of this is going well, and I've made quite a few improvements to it. It now takes less than half the time, just 25 minutes, in order to yield results. It's not as prone to carryover contamination, and it's easier to differentiate positives from negatives just by the naked eye. So this is one tool that I'm working on to prevent vertical transmission by letting us screen fish reproductive fluids for this pathogen in real time. But today I'm going to be focusing on another tool that I'm developing to prevent vertical transmission of psychrophilum. So we know that this vertical transmission is a big problem, and people do try to take measures to prevent it, particularly by surface disinfecting eggs using iodophor. And this definitely helps, but we know that it's not entirely effective. So the issue with the typical means of egg disinfection is that we know that psychrophilum is not only present on the surface of the egg, but we also believe that it can persist inside of the egg in this paraviduline space. And so the micropyle, which is this opening in the upper left where the milk or the sperm would come into the egg, that closes after the egg has been fertilized. So that means that disinfecting the egg with iodophor after fertilization, it might only remove bacteria on the surface of the egg, but not within. So what do we do to prevent this? And are we able to reduce intra-ova Flavobacterium psychrophilum loads? So in other words, can we in essence disinfect the paraviduline space? Which sounds kind of weird, but just bear with me for this. So first, I'm going to briefly explain how we typically disinfect fish eggs. So usually the eggs are artificially fertilized, and then they undergo what's called water hardening, where they firm up and they become a lot more resilient to handling. So it's during this step that eggs are usually surface disinfected with iodophor to hopefully kill or at least knock back any pathogens that might be present on the surface of the eggs. And like I mentioned earlier, this method of disinfection helps, but we know that it doesn't work perfectly because, again, psychrophilum can persist inside of the egg as well. So in order to get around this issue, what I've been working on is disinfecting the eggs before they're fertilized while the micropyle is still open in order to hopefully kill psychrophilum both on the surface of the egg as well as inside of the egg in the paravidalian space. So then after that, we would artificially fertilize the eggs. And this is a bit of a double-edged sword, and it's pretty unconventional because we want to get some of the iodophore disinfectant inside of the egg, but we don't want to damage them in doing so. So this whole idea is something that's already been piloted in Japan, and there's this study from Japan that lays the groundwork for my study. They did really great work, but we know that most of the strains of psychrophilum that we have in the US are distinct from those that are found in Japan. So if you've seen presentations from our lab before, you've probably seen this image, and I don't know if it will ever get less overwhelming to look at, but I just want to point out that this represents the genetic diversity of Flavobacterium psychrophilum, with each number being a distinct variant. So there's a lot of variation, and a lot of this variation is found in the US. And we also know that there are geographical differences. So again, we want to conduct a similar experiment to the Japanese study, but using US originating psychrophilum. And so to go back to this study for a moment, excitingly, when they disinfected eggs pre-fertilization, and then they followed it up with disinfecting the eggs after hardening, they didn't detect psychrophilum from the eyed eggs. So this is really promising and great work, but another component of this is that that we wanted to explore further is that they only took the experiment to the eyed egg stage for rainbow trout, but we know that bacterial cold water disease and rainbow trout fry syndrome typically present after a few weeks post-feeding. So we wanted to modify this work, perform it with the US psychrophilum isolate, and then also raise the resulting fry through at least a few weeks post-feeding. All right, so that brings me to my overarching hypothesis. So disinfecting eggs with iodophore prior to fertilization will reduce intra-ova Flavobacterium psychrophilum, thereby reducing transmission risk. So we decided to do this with rainbow trout and with a known rainbow trout associated and virulent Flavobacterium psychrophilum isolate. And then my primary objective was to evaluate the use of a modified iodophore-based egg disinfection technique to reduce or eliminate vertical transmission of egg-associated Flavobacterium psychrophilum. Okay, so in order to actually address these objectives and to test it, there's a ton of logistics involved. So one huge component of this is that we don't have a spawning population of rainbow trout at our lab. So thankfully, there is a hatchery that we work closely with that does have a spawning population, and we even have a health history for these fish too. So after having fish located, I then had to figure out how I was going to drive all the way there, which is a 4-hour drive, collect the eggs and sperm, carefully transport them back to the lab, while also coordinating growing enough bacteria and organizing everything else that we needed to do on the lab side of things. So going into this, I knew that it was going to be a really long day, even if everything went perfectly. And to make it even more challenging, this is dependent on fish spawning. So I only have a couple of tries each year to make this happen. So because of everything that would have to go into this, I figured it was probably smart to do at least a couple of pilot experiments to make sure that everything would go smoothly. So the first pilot experiment was just to ensure that the eggs would still be viable after transporting them across the state unfertilized. And I was able to successfully fertilize and hatch these eggs. So that one was a success. And then the second pilot experiment, I added a bit more complexity, and I wanted to verify that I could infect the eggs with psychrophilum and then recover it from them, and also pilot the pre-fertilization disinfection. So I was able to induce an infection in these eggs, and while it wasn't replicated, the principle of pre-fertilization disinfection did seem promising. So with this, I decided to move forward with the full experiment. All right, so on this slide, I'm showing the experimental design for the experiment. So I know that it's a lot to look at, so I'll try to take you through it. So on the left-hand side, I think that I have my pointer up yet. So on the left-hand side are the 6 tanks of 200 eggs that were not exposed to psychrophilum. On the right-hand side are 6 tanks of 200 eggs that were exposed to psychrophilum. Orange text here represents the pre-fertilization manipulations of either iodophore, saline, or no treatment. And then in blue text, I have the post-fertilization treatments of either iodophore or no treatment. So as an example, in this first group here, this tank was first pre-fertilization disinfected with 50 parts per million iodophore for 15 minutes, and then it received 100 parts per million iodophore during water hardening for 45 minutes. And then this is in contrast to something like the one right below, where this one received 50 parts per million iodophore for 15 minutes before being fertilized, but it received no treatment during water hardening. And I also want to point out that all of this was done in duplicate. So for both the negatives and the positives, it was all done twice. And then I specifically want to point out, let me get rid of that pointer, otherwise it's just going to stick around. I specifically want to point out these two tanks, which represent the current standard disinfection. And this varies a bit depending on where you get your information from, but we went with 100 parts per million iodophor during water hardening for 45 minutes. So some variation of this is typically how people would disinfect eggs. And then these two tanks represent the modified egg disinfection method, and were disinfected only with 50 parts per million iodophor for 15 minutes before being fertilized. All right, so I promise we're almost done with this slide, but I also want to note that the pre-fertilization disinfections were done with 50 parts per million iodophor in 0.75% saline in order to make it an isotonic solution to hopefully ensure that the micropyle would stay open during the treatment, both to make sure that we can get some of the disinfectant inside of the egg and that we won't reduce the viability of the egg because the micropyle needs to be open in order for it to be fertilized. All right, done with that side. So here's a picture just to show you what this actually looks like in real life. So all of the tanks are covered in this opaque plastic since the eggs are light sensitive. Then on the left-hand side are the negatives, on the right-hand side are the positives that were exposed to psychrophilum, and then again, 2 replicates of each. So for each of these tanks, I screened at least 20% of dead eggs and fry by first collecting them, and then surface disinfecting them with 400 parts per million iodophor for 15 minutes. Because remember, I want to tell if psychrophilum is present inside of the egg or the hatchlings. And then they were rinsed, they were homogenized, and then streaked down to culture medium. And then these cultures were incubated for 7 days, at which point I extracted DNA and then from any psychrophilum suspects, and then I confirmed its presence via the Toyama 1994 endpoint PCR. All right, so in addition to screening the dead eggs and fry, I also took samples from each stage of development. So I took 10 eyed eggs, 5 hatchlings, and 5 swim-up fry from each group, which were all treated similarly to the dead eggs and fry in that they were surface disinfected, homogenized whole, and then cultured. And then, like I mentioned earlier, cold water disease or rainbow trout fry syndrome, it usually will manifest post-feeding or a few weeks post-feeding. So I wanted to make sure that I looked at these time points as well. So for post-feeding fry, I took kidney cultures, and then the final sampling of fry at day 86, I took both kidney and spleen cultures. And the reason that I switched to kidney and spleen cultures instead of homogenizing whole for these last 2 groups is because I wanted to avoid potential contamination from the gastrointestinal tract since they were not feeding. All right, so we'll get into some of my results now. So for the rest of the presentation, just know that statistical analyses are still underway for all of this. So these are really just my raw results. But here we're looking at the percent mortality based on how many fish made it all the way to the end of the experiment. So to begin, I want to point out that in all cases, the percent mortality was higher in the groups that were exposed to psychrophilum than the groups that were not exposed to psychrophilum. And in some cases, this clearly isn't statistically significant, but it's still interesting given that some studies have suggested that psychrophilum may have a detrimental effect on eggs. And so then if we look at the conventional disinfection method, we see a percent mortality of around 78%, which is pretty similar to what we saw from the modified pre-fertilization disinfection only group. So basically, this is what we were hoping to see. And now I'm sure that you've noticed, but in general, these are some pretty high percent mortalities. But as I mentioned, these eggs were transported across the state and they waited hours for before they were fertilized. So in addition to that, we didn't use any sperm extenders for the mill. So these really high percent mortalities are pretty to be expected. All right, so here's the experimental design graphic again. I'm just showing this as sort of a main takeaway before we get into the specifics. So thankfully, I detected no psychrophilum from any of the groups that I didn't expose to psychrophilum, which might sound goofy, but it's always a good thing when you're working with a pathogen that can really sneakily be transmitted. I did, however, detect psychrophilum from all of the groups that I exposed to psychrophilum, except for one, which is this group right here that only received 50 parts per million iodophor for 15 minutes before being fertilized and had no treatment during hardening. So now we'll look at all of this a little bit closer. All right, so here we're seeing the prevalence of psychrophilum from the live eggs and fry that were sampled. So each of those developmental stages. And just to remind you, these treatments that I've shown, or I have the treatment shown here in the gray box. So the leftmost one here is the 50 parts per million iodophor for 15 minutes before being fertilized, and then hardened in 100 parts per million iodophor for 45 minutes. The next one is saline before being fertilized, hardened in 100 parts per million. The next one is the conventional disinfection method, so just hardened in 100 parts per million for 45 minutes. And then the next one is this modified 50 parts per million iodophore before being fertilized only. These other two groups did not receive disinfection. So I have it grayed out, but I still want to point out again that no psychrophilum was recovered from my negative controls shown in blue. And then from the groups that were exposed to psychrophilum, we recovered no psychrophilum from either the conventional or the modified disinfection groups. But we did see quite a bit of psychrophilum from the non-disinfected groups, with 21.74% of live eggs and fry that I sampled having psychrophilum, and 12.86 from the other group. So interestingly, I did find psychrophilum from this group here, which is the group that received only saline before being fertilized, and then was hardened in 100 parts per million iodophore at a prevalence of 1.43%. So it's low, but this is notable because this is a very similar group to the conventional disinfection group. And then I briefly just want to show how this shakes out, the distribution between where I recovered psychrophilum, either from eggs or from fry. So for that saline and then hardened in 100 parts per million, I found it was only in the eggs. I didn't find any from the fry. While for the saline and then hardened but undisinfected groups, it was primarily in the eggs. And then for the one that was just hardened with no pre-fertilization treatment, it was primarily in the fry. Okay, so now we'll look at the prevalence of psychrophilum from the dead eggs and fry that were collected and cultured daily. So here we see that in the conventional disinfection group, 5.06% of dead eggs and fry that I screened had psychrophilum, while 0% of the eggs that I screened, the eggs and fry that I screened from the modified pre-fertilization disinfection group had it. So then for the undisinfected groups, we also see almost 50% and almost 40% of the dead eggs and fry that I screened had psychrophilum. So really, if you take nothing else away from this whole presentation, just take away that it seems like it's pretty important to disinfect your eggs. And then again, I'm showing the distribution of where the psychrophilum was found within that prevalence. So, for the conventional disinfection group, for the dead eggs and fry, it was 50-50. And then for the saline and then hardened but undisinfected, it's similar to the live sampled groups. So primarily from the eggs, and then same for the group that had no disinfection at all and no pre-fertilization treatment, primarily found psychrophilum from the fry. All right, so to wrap everything up, horizontal transmission of psychrophilum is definitely important, but it's becoming more clear that vertical transmission is at the heart of a lot of this disease. And at least that's what we're seeing firsthand in Michigan. So this is why I'm focusing on vertical transmission. And as I mentioned earlier, last year I talked about the LAMP assay that I'm developing, and I'm hopeful that that will be a really valuable tool for preventing vertical transmission. And I'm hoping the same thing here with this modified egg disinfection method. So I was able to expose psychrophilum, or expose the eggs to psychrophilum and induce infection, and I was able to apparently inhibit or eliminate the infection in some of the treatments. So there's definitely more work to be done to identify if this might lead to differences in % IUP or % survival. But so far it does appear promising for reducing the incidence of Flavobacterium psychrophilum vertical transmission. So if this goes well, it could greatly improve fish health and reduce psychrophilum and associated losses. All right, so with that, thank you so much for having me. Thank you to the USDA NIFA for being the funding agency, to everybody involved in this project, Michigan DNR and Odin State Fish Hatchery for providing the reproductive fluids needed for this work. Of course, my advisor, Dr. Thomas Locke, and with that, I would be happy to take any questions.
Great, thank you very much, Sean. Those are impressive early results. Exciting to see where that's all going. Folks, feel free to drop your questions into the chat. We've got plenty of time, or you can unmute yourself and ask Sean directly. And we've got one here in the chat from Dave Groman wondering if, did you pre-treat at higher levels, like 100 parts per million?
So the original Japanese study, they did different disinfections at 50 parts per million, as well as 100 parts per million. There might have been another one that I might be forgetting, but they didn't really see much of a difference between 50 parts to 100 parts. So we selected 50 parts per million for the pre-fertilization disinfection, because our thinking was that, ideally the less disinfectant that you can use and for the shortest amount of time, while still being able to remove psychrophilum is probably best just for the sake of the eggs. So yeah, we didn't do any pre-treatments with greater than 50 parts per million, but that's the reasoning behind that.
Here's another question in the chat. Do you have a way to potentially produce large volumes of 0.75% saline for production hatcheries?
I'm sure that there is a way. I don't personally have a way. The way that I did this was basically like I think it was like a 12-liter carboy that I threw a stir bar into and very precariously balanced it on the the stir plate, and was like holding it while I'm pouring this massive amount of salt in there and just like hoping that it would dissolve and that the stir bar would stay where it was. So yeah, I don't have a specific solution to that, but I'm sure that there's a way to get around it.
Great big stir plates.
Yeah.
Another question here in the chat. Was there any record of higher egg mortality for the disinfected eggs compared to just the hardened eggs?
In general, so that's what I was showing a few slides back with the percent mortality. Really, the biggest difference that we saw was from the pre-fertilized, or not the pre-fertilization, the not psychrophilum exposed groups of eggs versus the ones that were exposed. It seemed like there was a little bit lower percent IUP for all of the groups that were exposed to psychrophilum. And again, none of it's, well, most of it probably isn't statistically significant, but we did see a bit of lower percent hatch from those ones that were exposed to psychrophilum. In terms of the actual treatments, it really depends, and there was a bit of difference with the replicates, which that's why we do replicates, but in general they were all pretty comparable.
Great. Any other questions out there?
Hey, Sean, I got one quick. Great job as always. You guys are doing good stuff up there. Out of curiosity, and I'm sorry if I missed it, is there any influence of temperature on efficacy and whether or not you have to alter these your treatment recommendations based on water temps?
I think for the most part, so what we did, I mean, all of this is taking place, like all of these disinfection manipulations are taking place before the eggs are placed into their tanks where they're, incubating and they're eventually going to hatch. So in general, what we were thinking is that the goal is just to keep all of the treatments, so the iodophore and the saline and the water that we were using to harden, similar to what they're eventually going to be put into in those tanks, just to avoid stressing out the eggs. I guess I don't specifically know if there would be any difference in the efficacy based on temperatures. There definitely could be, but we were more so concerned with not overly stressing the eggs out, especially given that they were already being raised in not ideal conditions after having to travel so far and wait so long before being fertilized.
Well, no doubt, sometimes that can be academic. You got to match the field conditions, and that's probably the most important part. So
yeah,
no, great talk.
Thank you.
Hey, Sean, I got a question for you. Hey, great talk. This is interesting stuff. Maybe I missed it, but after you pre-treated with formalin, how much did you have to wash the eggs out of the iodine, and then like how many washes did that take? And I guess my second question is there really any evidence that like a lot of biologists do water hardening out in the field with iodine during or post, like during fertilization or post fertilization? Is there any evidence that actually does anything? The way I look at it is just a double whammy, because then you take it to the hatchery and then you get treated again with iodine.
Yeah, so the first question about the rinsing after iodine. So yeah, for all of these, I generally tried to do, or I didn't generally try to, I did do 3 rinses after each treatment, including like the ones that just got saline. So 3 rinses of at least a 4-to-1 ratio of the egg volume to whatever it was that they were being treated with. And I did these rinses with saline for everything that occurred before they were fertilized, just to ensure that the micropyle would stay open. And then what you were saying about a lot of times they'll be hardened in iodophore after being fertilized in the field and then get it again once they're at the hatchery. To me, I think that doing it in the field is probably beneficial. One, from a standpoint of you don't want to be bringing any pathogens from the field into a hatchery or a facility. but also if you're, the micropile closing, it's not instant. So I think that doing disinfections during water hardening as opposed to hardening them and then transporting them is probably getting a little bit into what I'm trying to demonstrate in the pre-fertilization disinfection of hopefully trying to get a little bit of the disinfectant inside of the egg. So I would think that doing it during hardening immediately after fertilization probably gets at that a little bit, and I would think would have a better impact than waiting until they were at the hatchery.
Okay, thanks. And you don't see anything where the iodine that's within the micropile is affecting the milt at all, the sperm, right? I mean, obviously you didn't.
Yeah, I mean, not as far as we're able to tell. Like I said, the hatch rate between all of these, it was low, but they were raised under not ideal conditions. But the fact that the groups that were not exposed to iodine whatsoever, they had fairly similar hatch rates compared to the ones that were. To me, that would indicate that it likely doesn't have an effect on the ability of the milt to actually fertilize the eggs. But still more work to be done.
Right. Great job. Thank you.
Thank you.
All right, well, thanks a lot, Sean. Virtual round of applause for you too. Thanks for presenting today.